Okay. These questions then, these guidance questions, the reason it takes 4 sides is I've done it in large font. It was really to sort of leave you space for making a few notes on there. And it was also to fill it out to 4 rather than 3, which always annoys me. You've got a lot in this exam. All right, there's no getting away from that. It is fairly substantial, shall we say. So let's have a look at the unit reference sheet to start with. Don't need the end bit, do we? Because we're really only looking at the first 5 learning outcomes. This is not quite the same as what you've got in your handbooks. I don't know if anybody's still got their handbook even, but the unit reference sheets are in there. But in the course of the year, I requested a minor modification to it, assuming that I wouldn't be able to use it this year, and that would be from September, because there are certain dates in the year when you can modify units for the next run. But I asked, I think, back in March, and they just sort of passed it round, okayed it, and said, right, yes, that's fine, we'll put the new version on the Carver database. I thought, okay, right, we'll use the new one then, because nobody's going to know any different. So yeah, there are very slight changes. I can't remember exactly what they were, but it was when I started trying to draft an exam, I realised that actually making the questions work required a slightly different arrangement. So what we have, the first learning outcomes about cell division, and so you'll have essentially 3 questions. 1 on the replication of DNA, 1 on the main stages of mitosis and or meiosis. Tends to be mitosis in the pass section. All right, I might ask about meiosis features in the merit distinction bit, but for the pass section it's just mitosis. And then a comparison of mitosis and meiosis, you know, just how many divisions is there crossing over, how many daughter cells, that sort of thing. All right, so 3 questions under the cell division section. The control of gene expression, yeah, this was not my finest hour, this one. The bit on stem cells is really just about saying what the types are, you know, totipotent, pluripotent, multipotent, that sort of thing, and what their key features are. And then explained, which you're not actually really going to explain, it's only really going to be state, I suppose, examples of processes involved in the control of gene expression. And it'll be things like, you know, well, what's estrogen and what does it do? You know, it's a steroid hormone, it gets into the cells, it combines with a transporter, attaches itself to the DNA, and it controls transcription. That's it, it's a transcription factor. So you don't need to understand the mechanism, that sort of thing. Epigenetics, you know, I'm not going to ask you to explain that Waddington's landscape or anything like that I showed you on that PowerPoint, but just what are the 2 main processes? And it's DNA methylation and histone acetylation. That's it. You know, I'm literally giving you answers. So it's not a big chunk. So if that did seem a bit vague, yeah, don't worry. There's not a lot in the questions, and I think that was my problem. The next one, I think I collapsed here. There was only, sorry, there were 2 assessment criteria to do with DNA probes, and I couldn't actually make 2 questions out of it. And it was really about the DNA probes are the single-stranded bits of DNA with the matching bases for a sequence you want to find. You know, and it's like having a jigsaw where you've got a definitely shaped piece and you're given the piece that fits into it. That's the gene probe, the bit that will actually pair up. It'll base pair with the bit that you want. If you add a fluorescent label or a radioactive label, you can then find that bit. So you can diagnose human diseases, anything caused by a gene malfunction or mutation, or you know, just a particular allele. You can find it using DNA probes. The hybridization is just when your probe binds to the target sequence. That's it. You know, there's nothing magical about that, really. It, yeah, it's quite straightforward. You then have a fairly big chunk here about inheritance. So solving problems involving Mendelian and or non-Mendelian inheritance, so things like codominance and that sort of thing. So questions sort of like the blood groups scenario, you know, what phenotypes could you get from what genotypes and that sort of thing. And then a problem relating to population genetics is a Hardy-Weinberg calculation, and it does require you to understand a little bit about genetic drift. And I think if you get your head around that bit, and I was quite pleased with, you know, what I'd put in on that, the idea particularly of the founder effect, that you know, if you understand that, then this will make a little more sense. And then the last bit is really what we've done over, well, this week, isn't it? Principles of variation and natural selection to explain the evolution of new species. So just, you know, what does variation come from? Well, genetic mutations. And then just explaining a couple of little examples about natural selection, one of which is directional, the other is stabilizing. All right, I won't put the disruptive one in. And then just a little bit identifying, I think, you know, the allopatric and sympatric aspects of speciation. And then last bit is about the taxonomic ranks and the principles of nomenclature, which is pretty much what I showed you on the end of that PowerPoint, where, you know, I'll leave some gaps on there. Genus and species would come from a name, and other gaps you've just got to remember the order of the ranks. Okay. The other bit on here, of course, is the presentation. So there is a lot there. You know, that's what, 3, 4, 5, 6, 7, 8, 9, 10 questions in the pass part. So it's one of the biggest exams, this one. I am a bit concerned about the size of it, but yeah, I mean, if we find that everybody's rushed and, you know, struggling to finish it, then I will have to allow for that. The problem, I think, was because there's so much in the unit, obviously it creates a lot of past questions for a start, but then because some people get on better with some areas than others, I wanted to pick questions from several parts of the content to go in the merit distinctions section, and that ended up making it really big. I think perhaps what I should have done, which I haven't, and it might be too late, I should perhaps, I don't know if I've put, so let's say there were 4 questions in the merit distinction, I should have said, you know, choose 3 from 4 or something like that, which is typical of what you get at university. You get to make a choice about which questions you answer. The trouble is, you can find you spend so long trying to work out which ones to answer that you might as well have answered them all anyway. So, yeah, hopefully it won't cause a problem. If you're well prepared, then, yeah, everything's so much easier, isn't it? Is what? Oh, the modified URS. Good point. I will put that on Moodle. Yeah. So it's a little bit clearer then. We've got a mock exam as well to come up, of course, as well. Excuse me. But what I would suggest you do, I can't remember whether I said this the other day, you know, with the idea, oh, it was yesterday, wasn't it? Yesterday afternoon. I felt that didn't seem the right time in the week. If you found the idea of the open book exam quite good and that production of the notes quite a useful exercise, still do it. You know, the fact that you won't have the notes in the exam is you know, it's another point altogether. But if you go through the process of taking your whole unit's notes and then trimming it down and then trimming it down and trimming it down so it fits on perhaps, you know, perhaps 3 or 4 sheets of paper or something like that, use those as your sort of final revision materials, you will find that you are doing something very useful, which is actually more useful than having the notes in the exam. But you are processing the information and trying to make sense of it in a way that you can remember or you can use. So, you know, I would suggest that as an idea. Right, let's just have a look through. I'm not going to sort of give you all the answers and things to this yet. What I want to do, try to think when we have got another bit of revision time, haven't we? But not very much. We're we'll have a quick go through, just go over these and make sure that you're clear on what they're referring to. And then I think we might have a look at one of the A-level questions on evolution and speciation, perhaps, because we do not have a lot of time for revision. All right, and it's because we lose all this chunk of time for the exams and things, doesn't it? But there's time on the plan. 2 weeks today, I think. We've got revision and the mock exam, haven't we? Or is the revision before that? No, there isn't, is there? That's mock exam and go over the mock exam. Is there a revision after that? I can't remember. The whole plan is not committed to memory.
If we had time, what if we were, say, trying to make 4 slides of it,
Yeah.
2 pages, have that for a mock exam?
Yes, yeah, that's a good idea actually. Yes, I think, yeah, if we say that the mock exam is an open book in the style of a few sheets of paper, and that's right. Yeah, I think. Yes, I think that's a really good idea. That because I think yeah, so your mock's in 2 weeks, and then the exam is another 2 weeks after that, isn't it? I think. Oh, is it the week after? The week after the actual mock. So is it 3 weeks till the 1, 2, 3 weeks till the mock, and then the exam is the week after that. But have we got another revision session? We must have on the Tuesday afternoon, I think. Tuesday afternoon before the exam. We've got revision. Yeah, we've got. Right, yeah. Right, so we have got a revision session of a couple of hours. So that's, yeah, I couldn't in my mind, because I hadn't actually committed the plans to memory yet, couldn't in my mind remember if we'd got a revision session, which, yeah, that's a relief, because that means, so that is literally the day before the exam, or before the mock. Before the exam. Oh, that's about as good as you can get then, isn't it? You know, because you can then be asking the questions. Yes, so I think in terms of preparing for the mock, then yes, I think that's a really good idea. Condense your notes into a bigger version of what you had for the open book exams. So yeah, you know, for some people it might be 3 sheets, for some it might be 4 or 5, you know, but getting it down to what you think you will need. And then if that material is useful in the mock, you know it will be useful in the real one. All right, now I will give you more guidance on the merit distinction bits when we get a bit closer, but I want people to be secure on the pass bits before anything else. Yeah, because you know, when all's said and done, that's the bit that's going to make the biggest difference, isn't it? Okay, you've got then learning outcome 1. We've got these 3 sections. So learning outcome 1 is the cell division bit. And 1.1, what I'm going to do actually, I'm not going to start a new slide on the board. I'm going to go right back to the beginning and just pick through some of the things that are important on here. The first bit about DNA replication, I don't actually think it's on here. No, it's not. The replication bit, all I have on the noteboard is on there. Yes, the other is on PowerPoint. So what I would suggest, so let's, I know where it is. Find you the bits that you need. It's in the cell division one. All right, the bit that you need for the replication is, the background is here, but that's very simplistic. And you can see from the questions I'm asking you, what you need is that. All right, so that is slide number 6, if I've put them all in. So leading strand, lagging strand, helicase, DNA polymerase, DNA ligase, and the Okazaki fragments. They're all on there. All right, so it's, and I'm going to give this away. Basically, it's that diagram that you'll need to be able to label and say what the enzymes do, and you know, this Yeah, not too complex. Actually, that's a more complicated version because I gave you the one here with those proteins on there and the topoisomerase. So you don't need all of these that are on here, all right? Just the ones that are listed on your question sheet. Probably yes. Yeah. I think all the PowerPoints I've used are on Moodle. I will check. If you find any that are missing, let me know. So that particular slide is useful for that. Moving on to phases in mitosis. Move on. There's none of this stuff about bubbles. Mitosis, you've got the significant features of interphase, prophase, metaphase, anaphase, telophase. It's being able to recognize diagrams not unlike this. There are loads of them out there, and you can easily test yourself on these. If you search for mitosis diagram, you'll find, in fact, if you search for mitosis diagram unlabeled, you actually get sort of blank ones to put the labels onto. What you actually want, though, are diagrams that are perhaps in the wrong order, and you've got to reorganize them so that you get an idea of that sequence, that the chromosomes are not visible in interphase, but then they become visible, then the nucleus disappears, they line up across the middle, they get pulled towards the end, then you get 2 new nuclei, and it's just that, organizing them. Or, you know, you could find a set of diagrams and print them off, cut them out, and then, you know, play at rearranging them. You know, don't put the labels on them necessarily. You know, write those on the back and then rearrange them so they're in the right order. So you can see what the chromosomes are up to. So if you go around this one, no visible chromosomes, then you get to prophase, they start to become visible. And so by late prophase, they're there, but they're not organized in any way. In metaphase, they're across the middle. Anaphase, they're pulled apart. Telophase, you get 2 new nuclei. And then, you know, you could put one of these round again if you wanted to. So that's the first bit of 1.2. I think, hang on a minute, that should be then, right, I'm just having a look. Yeah, the way I've laid this out is wrong, isn't it? Instead of being 1.2a and then 1.2b and c, 1.2b should actually be 1.3 on the new. I don't think I changed that, but 1.3 is actually comparing mitosis and meiosis. In terms of the number of divisions, so is it 1 or 2? The purpose, is it for body cells or gametes? The number of chromosomes, is it the same or haploid? And the contribution to genetic variation is, trying to think whether that's in the past section. I think that's in the merit distinction. Part C on this, that doesn't actually necessarily belong in any particular bit, but it sort of goes with the words that you need for this section. All right, so part C over the page there is general, perhaps. Haploid being single set of chromosomes, diploid being pairs, crossing over is the exchange of parts of homologous chromosomes in prophase 1 of meiosis. Independent assortment is where you can put, you know, any combination of chromosome 1, 2, 3, 4, and so on. But as long as you've got a full set. But that, it's sort of embedded in, really, rather than being direct questions. Okay, so I think, yeah, that's all there is in the past bit. I'm not asking about, oh, yeah, you got the comparison bit. There's lots of stuff on there. The next section is then about stem cells. And again, the thing is, because a lot of this is really heavily diagram based, you've got quite a lot of PowerPoints. I wasn't sure whether you'd got, did I give you this one? I did, didn't I? All right, so the different types of stem cell. well, characteristic features of stem cells, sorry, is the first bit, and that's those two things: self-renewal and differentiation. So the fact that they can produce more of themselves, but they also differentiate into specialized cells. And then describing the following types of stem cells. I'm not sure they're all in here. I'm not sure whether we've got the keep going, whether we have the totipotent ones on here. If you read the notes that go with it underneath, yeah, they're important to look at those. Yeah, all right, we've got embryonic stem cells are here. Are they described as being totipotent? No, not on there, they're not. Right, they're described as pluripotent because they are at that stage. It's only the first few divisions where you get the totipotent ones, and I don't know whether, I don't think I wrote any notes on here, did I either? Let's have a look. Doesn't look like it. Looks like it went straight onto the DNA technology. All right, so you might just need to, well, is it in? It's in there somewhere, is it? Oh, right, in the stem cell jargon bit at the end. Is it? Was it?
Well, yeah, you talk about it. And
Oh, right.
because I have all the cells in the
Ah, right.
embryos.
Yeah,
You mentioned
that was it. Because I remember talking about the embryonic membranes and things.
So was after, so it was the 90.
Right. Okay. So yeah, right down at the bottom
I'm not sure if it's
bit.
right.
I added it.
Yes.
I added it to that one. All right. So yeah, you may find you got that one, and we've added totipotent to that one because that it sort of comes in above pluripotent. It's not just all the types of specialized cells in the body, but these are the first few divisions in the embryo where you can get the membranes as well. We don't need totipotent cells for research and stuff like that. The pluripotent ones are very useful, and multipotent are still useful as well. Unipotent, which for some strange reason has got a square bracket in the middle of the word on your questions there. Uni bracket potent. Is the bracket? The bracket's next to the P. That's why. So yeah, unipotent, they literally can only be one type of cell, so that's like skin cells, for example. The induced pluripotent ones are the ones that are produced in the lab, where it could be, for example, bone marrow cells treated to switch certain genes on and off. So they go back to being like pluripotent stem cells, which you can then perhaps turn into heart. So, you know, you could turn blood into heart, but you have to wind it back first and then send it down a different route. Okay, now, trying to think where I would have gone from there. The next bit that we have on here, the recombinant DNA technology, yeah, that was mostly in your notes, and that is largely related to the assignment rather than the exam. So this bit doesn't really come into the exam. Well, not directly, I suppose. It could do with the gene probes bit. Try to find the, yeah, there's, right. I've got this in the wrong order. The DNA probes bit is actually 3.1, isn't it? So what's meant by that? Well, it's just this radioactively or fluorescently labeled piece of single-stranded DNA with a complementary code to a target sequence that we're looking for. So a little definition of it, if you like. And there's the control of gene expression. So yeah, I've taught them in the other order here, obviously, which is 2.2, estrogen being an example of transcription factors. the stem cells bit, yeah, I've just got them organised the wrong way around. The stem cells bit we've now just gone over. Transcription factors are things like, estrogen's an example, all the steroid hormones work like this, and I showed you the bacterial one, the lacoperon being the most famously known one. Just with this ability to switch transcription on or off, depending on conditions. So, I mean, with estrogen, for example, it's a classic example, of course, is female reproductive hormones, where you don't want them all doing the same job all the time, because it's really important that one switches on, and then the presence of that one switches another one off, but that one then switches a different one on, which switches this one off, and so it goes on. And so this idea of controlling the expression of different genes is then what gives rise to, the reproductive cycle. And it's just this binding to a promoter area where it can then control transcription of the gene. So it's a transcription factor. And the only thing about epigenetic control was just taking these two examples of how during lifetimes the genome, in terms of the DNA and the histones that it's wrapped around, can be altered, and that also changes the expression of genes. The RNA interference, this is where things went wrong, because I was a page of notes missing. So you have got those now, I hope, in colour. And you had this natural production of small interfering RNA molecules, which have this rather bizarre shape. To start with, they are this hairpin sort of shape. They bind to a protein, which is this RNA-induced silencing complex. That breaks the hairpin into two pieces. One's lost, but the other bit, and it makes you wonder why it was produced like that in the first place, stays attached to that protein. That then attaches to messenger RNA and chops it up into bits, so it's not used anymore. So, if you think about this, if you start expressing a gene, well, you only want a certain amount of messenger RNA, you only want a certain amount of protein from that, and so when you finish needing that, you need a way of switching it off. And so that's what this does. And of course, there is scope to use this as a form of medication to silence genes. So that's what this does, in a way, gene silencing. It stops the expression of a gene, and if that's a faulty gene that's causing, a condition in somebody, then yeah, you can stop the effects. Right. Oh, the other bit on there. How can cells use it to defend themselves? Well, theoretically, you can use it to chop up the DNA or RNA from viruses. I think our cells don't, but I think bacteria are quite good at that. They're quite good at defending themselves against viruses, to be honest. Right, what else have we got? We then went on to the inheritance bit. So the terminology here, it's not just a matter of learning particular words, but just being really clear about, what's a chromosome, what's a gene, the idea of alleles being different forms of a gene, being able to clarify, dominant and recessive as not being strong and weak. It's just a matter of how many copies you need for them to be expressed. So dominant, it'll be expressed even if you've only got one copy of it, but the other chromosomes carrying the recessive. Obviously be expressed if you've got two copies. Recessive, you only get that one expressed when you've got two copies, and that's the real game changer when it comes to identifying individuals' genotypes. In a population, you can only ever identify the individuals carrying two recessive alleles, the homozygous recessive. Codominant, both of two different alleles expressed, so something like the A and B blood groups, or the black and ginger in tortoiseshell cats. You also get dominance hierarchies, and the example on here, I think, yes, I've given you a hierarchy one to work out, and there is one like that on the exam paper. There's one on the mock as well, so you'll see what I mean. where you might have several alleles and there's a, this one's dominant over all of the others, this one's recessive to all of the others, but there's perhaps 2 or 3 others in the middle where you've got, well, this one's dominant over that one but recessive to that one, and yeah, it's not as simple as just a sort of on-off switch. So making sure you're clear on the terminology that goes with this, so that not only do you understand it in the questions, but you can also use it in the answers. The notation for these, using the uppercase and lowercase, really important. Remembering that any individual organism, if it's diploid, if it's the body cells that we're talking about, must have 2 copies of every gene, as long as it's not sex-linked. I haven't put a sex-linked example in because that does cause problems. So it'll always be 2 letters for the parent. But when you start to look at gametes, and what I didn't do in here was to label up the lines on. Oh, that was because that was just a particular question I was answering, wasn't it? How could 2 brown-eyed parents have a blue-eyed child? But at the top here, you've got the big B, little b's are the 2 brown-eyed parents, both heterozygous. The individual letters are the gametes that those parents produce, and you can then put those gametes together in any combination, and you either draw it like that or the square, which of course I then did wrong, didn't I, when we came down here. We did, the P colour one as a demonstration, and then I, for some bizarre reason, did the next cross as the square. I should have had big Y on one side, little y on the other, but hence the line I drew across there. And you've got this example, a gene for flower colour has the following alleles, and it tells you which ones are dominant, which is recessive, and I've made it easy, I've only put a 3 in there. Alright, I won't say any more about that. You might want to have a go at that while you're here. Then the next bit I haven't put, I have got multiple alleles on there. I haven't got anything sex linked. I haven't got a dihybrid cross. We definitely haven't got anything on the chi-squared test. That was just a quick recap, and I said if you need to use it, go back to the cells unit. Then we looked at the population genetics. This idea of genotype and allele frequencies being really important. They can be affected by migration, mutation, selection, and then this idea of genetic drift. Now, selection, of course, is what we've done in evolution. Mutation we did in the last unit. Migration is not something you can particularly model, not something that we can actually do as a quick thing. The genetic drift, we had the idea of bottleneck effects, where you've got big contractions in population numbers, which might really reduce the number of different alleles or genotypes that could get through to, to actually grow the population again. You end up with this reduction in diversity and increased inbreeding. The more useful example was the founder effect, special case of bottleneck when you get this small population, and you could have these founder populations which have either a high or a low incidence of certain alleles. And I introduced you to the Amish example. There is a question relating to that as a Hardy-Weinberg. All right, so it is, understanding a little bit about the founder effect and the influence that, the other thing that goes in there is lethal alleles. Because if one of these, quite rare in the main population, if one of these alleles that ended up in the founder population was lethal, you do get some interesting effects. And of course with lethal alleles, if they are fatal before birth or shortly after or in childhood, they don't appear in the adult population. So you have to bear that in mind. You won't always have the same genotype or allele frequencies in newborns as you do in adults. So make sure you're looking carefully at that one. And then we went on to the Hardy-Weinberg bit. So, this only applies if. Okay, yeah, and that obviously doesn't apply very often, but it just sort of had to be our best guess, didn't it, really? And we worked through the definitions of the letters, the P, the Q, the P squared, Q squared, 2PQ. There is a calculation to do. I've started it. So, because over the years I've discovered that people can't pull out that initial bit of information very easily in exam conditions for some reason. So I have started it, and it's then just continuing it on to get carriers. And then we went through the examples that we could find in the A-level questions. And that little bit there, observe Q squared, square root it to get Q, take that away from 1 to get P, and then do what you like to get the other things, depending on what you're asked for. That is the key, really, to getting Hardy-Weinberg. And those are the examples of the exam papers. That is probably it. All right. So that, just as a reminder of what's what. Now it's sort of up to you, really, as to whether we do anything else now or not. So we'll stop it at that point.